Everyone. My name is Allison Garcia. I'm a member of the team supporting the Biomedical Research Informatics Computing System under Dr. Matt McAuliffe at the Center for Information Technology. Thank you so much for joining us today. I believe this is the 5th installation of the seminar series in 2024. We do have a really exciting agenda for today's meeting. So I will quickly kind of go through the agenda and some logistics and then turn it over to Dr. McAuliffe. Today, we are excited to present to you a BRICS video. We also will be talking about the items that were just released as part of our release cycle. So some of the enhancements and new features and capabilities being brought to the BRICS platform. We will also be unveiling a new video that talks about the platform itself. And then we're going to transition and have a demonstration of 2 capabilities within the BRICS ecosystem, STAMS and CTMS. And then we're going to allow time throughout the session for folks to be able to pose questions. So please take advantage of using the chat feature within Teams. We will find appropriate times throughout the presentation today to hopefully pause and speak to the questions in the chat, or people can use the raise hand function. If not, we will definitely reserve some time at the end. Please be aware that today's session will be recorded. Please keep your microphone muted and come off mute at appropriate times when asking questions. We always post the recordings for folks to be able to come back and learn at a future date, or for folks that are unable to attend the session. So it's posted to our public website at the URL on the screen here. But definitely, we want this to be an interactive session and encourage your participation. So with that, I will turn it over to Dr. McAuliffe to kick us off. Thank you.
All right, good morning. So usually I start off just so everyone has kind of the broad view of what BRICS is and the kind of general functionality and how it can be deployed in different ways to meet making data fair, findable, accessible, interoperable, and reusable. But instead of me showing a few slides and discussing it, we recently worked with the CIT comms group to put together this video that is now also posted at the BRICS website. But it's about a 4-minute video, and it kind of gives a good broad overview of the kind of work that we're trying to, or the kind of work that we're doing to support, again, making data shareable. So with that, Preeti or Allison, if you want to go ahead and start the video, that'd be great.
What if I told you there was an easily customizable system that could help manage your data across the entire research lifecycle? The Biomedical Research Informatics Computing System, or BRICS, does just that. Developed by the National Institutes of Health to meet the unique needs of life science researchers, BRICS is an enterprise solution for end-to-end research support and is a customizable data science platform that simplifies data management and helps researchers comply with the NIH Strategic Plan for Data Science, making their data more findable, accessible, interoperable, and reusable. At a high level, BRICS is a web-based system comprised of 12+ modules that work together to efficiently collect, validate, harmonize, analyze, and share research datasets. BRICS supports all data types, including clinical assessment, imaging, omics, biosample, and preclinical, and can be deployed both on-premise and in the cloud. BRICS helps harmonize data by using common data elements for efficient analysis and collaboration across studies. The best part, you can mix and match the modules you use based on your specific needs, whether that means using all the modules or just a few. So what does using BRICS actually look like? Let's take a look at an example of how a researcher manages their data and unlocks data exploration using the Data Dictionary, ProForms, Data Repository, and Query Tool modules in BRICS. First, the researcher uses the Data Dictionary to easily design a data upload form that uses common data elements to standardize collection and facilitate rich cross-study analysis in the future. Next, the researcher collects their data, For streamlined live data collection within BRICS, they can use the ProForms module, which offers near real-time data validation. Data entered via ProForms is validated, uploaded, and stored in the data repository module as it's entered. ProForms is 21 CFR Part 11 compliant to meet FDA requirements for clinical trials. ProForms is highly flexible and able to accommodate a wide variety of data collection methods. In addition, researchers can use their own data collection tool and submit their data into a BRICS system. When the data is submitted to the data repository, either via ProForms or manual upload, it's validated to make sure each value aligns with the accepted common data elements. This extensive validation process helps standardize the data in the BRICS system to enable efficient cross-study analysis across hundreds of thousands of subjects and counting. Once the data has been collected, validated, and stored, it's time for the exciting part: data exploration and analysis with the query tool. Researchers can use this tool to query across studies and data types, enabling them to expand their research cohorts, visualize aggregated data in more meaningful ways, identify patterns across studies, diseases, and regions, and collaborate more effectively with other researchers. This is just one of the many ways researchers are using BRICS, and as the needs of researchers change, the system will continue to evolve, driving data science and collaboration forward and delivering innovative, powerful new tools to the data science community. Hundreds of NIH and NIH-funded research studies are already using BRICS to turn discovery into health. Ready to join them? See how BRICS can help you shorten the path from data to insight by requesting a demo today.
All right, so with that, I think I'm handing it off to you, Preeti, to give a demo and talk about some of the new functionality that we just had with our Spider-Man release earlier this week.
Yes, good morning, everyone. I'm Preeti Roy. I work as a PM on the BRICS project with Matt. And like Matt mentioned, we recently had a release. Our releases are named after superheroes. So the last one that we had was Spider-Man release. And we implemented quite a few new features across the different modules. But I'm just going to go over main highlights. So I'm using the Fitbit instance for doing the demo. And as most of you are familiar that we moved on to using RAS for authentication. And in the past releases, we were supporting NIH PIV card and CAC card. With this release, we also started supporting the VA card. So all the updates, you can see it on the login page in here as well. I'm going to start with the meta study module. In here, when you start creating a new meta study, we introduced like the study state. You can select meta-analysis or prospective or retrospective based on the study type or the data that you are planning to submit for the meta study. And in the light of NIH's data sharing and management policy, we also introduced different data storage under the meta study. So we came up with different types and categories that you can use for uploading the data from any external NIH data research that you have performed. So you will be coming to this tab and uploading the data in here. And the different types of data that you can upload, they can be classified as biosample, clinical assessment, genomics, imaging, etc. And to go with these, we also have categories. So you can associate the different categories with the different data types in here. So besides all these, we also introduced the filters on the left pane to go with the new newly added fields. So you can select by the different study sets in here, data types, and data categories in here. We also introduced a new capability where when you create a new meta study, and if it's in draft status, we will be minting the DOI, which is in the draft status. So you don't have to ask for a new DOI to be minted when you're creating a new meta study. So those are the enhancements for the meta study. In the accounts management, again, it's a lot of like UI changes that we made. So all the tabs that you see in here, they used to be accordions earlier. So the user had to scroll down a lot and go to specific sections to see things. Now we have like organized them in the tabular format. So it's easier to just go through the tabs and see all the information. So this change, it's been applied across the different sections of the page. Under the account list, we did the same. If you select any user, you have, again, the tabular status and structure in there. And same for the account reviewer and the different dashboards that we have in here. We also did performance improvement across the site, especially in the dictionary. If you go under the data element and manage, this page used to take a while to load. It's become like much faster. And if you perform a search in here, that is also returned at like a much faster pace at this time. Same for the form structure. So if you do a search in here, this also used to take a long time, and now you can see that it returns really fast. Another big change that we have done is in the query tool. This module has been transformed from the UI perspective. We did a lot of changes with the look and feel. The left tabs that you see, we kind of worked on these aesthetically, as well as like earlier it used to be a bit confusing for the users. We used to display studies in here when forms were selected. So we streamlined that, and now you can see forms when you select in here, studies. When you go in here, you see the studies in here, and you can search for the forms in here. The right pane that you see, we have adjusted the spacing across the different boxes in here as well. So the users, they can see more data when you come to this page itself. Another big change that has happened was around the data cart and the join form view. So if I search for a study in here, add the items to the cart, we have two different views now. So data cart view is for someone who's not trying to do the joins and just wants to take a look at their data and just download and get out of the system. So this is the view for them. They can come in here. They don't have to try to do any joins or anything. They come in here, see the data, download it, and they're done. Join forms, this is for someone who wants to perform joins across multiple form structures and see data as combined view. So for them, I don't know how many of you are familiar with the system. So earlier we had drag and drop feature. So we used to take the forms from this and used to drag on the right pane. We used to have another box in here. So we could join like up to 5 forms. So that was like a bit of inconvenience. So we changed that flow as well. So now you can select the forms that you want to join through the checkboxes. And they show up in the top, and you can, again, like select up to 5. If you want to move the forms up and down, you can do that as well. And then you can perform the join in here. For the download, we also made some changes in here. So some of the forms, they have files attached to them. So by default, when you do the download, you will get the associated files. But if you choose not to get the associated files or attached files, and if you're just interested in looking at the data for the file, you can uncheck this and then do the download for it. I'm not going to do the download, but you get the idea. Yeah, and we are continuing to work on Globus integration, which is like a feature where large volume of data and associated files, they can be integrated with the submission tool, and the users can use that. And we are also continuing to work on MongoDB integration or migration with our system, which will help us with unifying the data that we get from Proforms. It will be available in repository at the real time. So that's all at the high level that we implemented. There's multiple other features, but they are available on the release notes. If you are interested to take a look at them, it's available on the BRICS website under the release notes. And I also have it added to the slides in case anyone is interested in reading about more of the features. Any questions?
Great demo. Thanks, Preeti. As you can see, we're constantly trying to improve the usability and performance of the system, and we continue also to add new functionality as we release new versions, which again, we release probably 3 or 4 times a year. But if there are no questions at this time, we can move right into Dr. Nathan's talk on STAMS and clinical trials management. So with that, we'll hand it off to you, Dominic, but please feel free if you got any questions or save your questions to the end for Dominic, but it's all yours, Dominic. But you're on mute. Okay.
Thank you, Dr. McAuliffe. I'm going to present here. Okay, is my screen visible?
Yes.
Right. So thank you for hosting me. My talk today is going to be on the STAMS module, which is the Specimen Tracking and Management module. Disclaimer, if I say anything bad, it's all my fault. Don't blame the government or any other groups. It's all me. BRICS consists of a set of different modules, and all of these modules interact with one another, and they provide lots of rich features to the platform. The module that I'm talking about today is STAMS, which is the Specimen Tracking and Management module. STAMS is used to manage biosamples or biospecimens. For example, any fluids, tissues, if you have blocks, slices, slabs, if you pull out a tumor tissue, those samples, and it can also manage brain tissue extensively. STAMS is also used to inventory physical freezers and storage racks. So if you have a freezer farm with a lot of samples and you would like to keep track of that, this is what STAMS can do. One of the key features of STAMS is being able to integrate information from data that has been collected either from clinical studies or research studies and have everything in one integrated system. So you do not have to depend on multiple siloed systems. We have this full features in one system. So with the samples, you can also match those to subjects. You can match those to protocols, diagnoses, and any other such as imaging or genomics data that you may have. STAMS also allows the management of users and role-based access. So as I mentioned before, STAMS pulls data and integrates with data from Proforms, which is used extensively for protocol data collection. And it also integrates with the medical record system at the NIH, which is called CRIS. And CRIS actually has a surrogate data warehouse called BTRIS. So any medical record information, any clinical assessment, any treatment information, all of those are pulled into STAMS directly. And the key highlight here is we have the flexibility of integrating, because we have already done this at the NIH. If we were to port the system out, we have the capability of integrating with any medical record system that is electronic. On a basic structure, STAMS offers the flexibility. So we can define a repository and then a freezer. So we can have any number of freezers. And within the freezer, we can set up the freezer structure. So this can accommodate boxes, racks, cassettes, or any other like freezer format. On a very simple layout is the box setup. And the box setup allows you to define what size of box do you have, how many rows, how many columns, coordinate formats, either alphabets, numbers, or combinations of the two. We also have the ability to define what kind of samples are being collected. We call these cell types. So in this example, we have blood, brain tissue, cerebrospinal fluid, plasma, saliva, and also urine. We can also specify what kind of cryo solutions are being used and what kind of units are being used for each of these samples. We can also attach diagnoses to each repository. This is a simple example where we have 3 ICD-10 diagnoses, but there are other repositories with far more extensive diagnoses. So these can be customized based on the repository or the disease or the research that's being conducted. We have the ability to add subjects. So one important thing I would like to highlight here is there are 2 buttons. There is a add patient and add animal. So STAMS is flexible enough that you can also use it for preclinical studies. There are some studies that have extensive fruit flies, mice, and also ferret samples that they are managing, and you can use STAMS to track those systems. If you were to add a patient, we can pull medical record information such as demographics, address, phone number, age, gender. We can pull that in directly from the medical record system. Or if you have a standalone repository where you're just managing samples, you can add subjects in manually also. And we have the ability to anonymize subjects, as Dr. McAuliffe mentioned before. You can generate a GUID, so you can partition out identifiable information and have it fully anonymized, or if you need it for clinical purposes, then you can have fully identifiable information. This is just an example of adding a sample into a freezer box. So in this example, I'm adding blood. I would have the sample type, which is blood, a description, and then what is the cryo solution. Next, I will also have the protocol that this is associated with, the patient name, medical record number. If the protocol is anonymized, then there would not be, the patient name will not appear, but there will be something in place, which would be either GUID or subject ID. I can also include diagnosis of patient notes from clinic as needed. And then I have the time that the sample was collected. It keeps track of who has collected it, the time that it's frozen, who has frozen it. I also have other fields such as the barcode, which the system generates, and then the position in the freezer. And the current value, I've collected 1 milliliter of blood. And if there's any procurer information, notes, and I have all these tracked. Lastly, I can also add diagnosis information. And as I mentioned before, we have the ability to pull from medical records directly in here. What is also important is all of these fields can be customized to be printed out onto a barcode, onto a label printer that then can be attached to the sample. I can also track distribution. So if I were to give samples out to collaborators, I can track when the shipment is done, how much I'm giving out. And here, as I do this in real time, my sample, the current volume originally was 1 milliliter, now it goes down to 0.9 milliliters. And I also have a full audit trail that shows me what has changed in the sample and what has where has the sample gone to. I can also aliquot samples. So if I have a parent sample and I would like to have 3 aliquots, I can specify how much each one is. And then I can also do this. So this is my parent sample, and I had 0.9 milliliters. If I do 3 aliquots of 0.1, now it's down to 0.6 milliliters. And each one of these would show this is the source patient, medical record number, and then the test protocol. And it also has information on the parent sample that this was obtained from. And it also has, what is the remaining samples. So if I click on the parent sample, it will show me the audit trail. And again, I can go back and check. And this is important for inventory purposes, especially as your repository grows and the number of samples grow. I also can't search for samples. If I have a really large repository, the STAMS platform is compatible with barcode scanners and also label printers. So if I have a barcode scanner, I don't have to type in each barcode or each sample. I can retrieve the sample from the freezer, scan it, and the barcode would actually appear in this red highlighted box here. And all you have to do is say search inventory, and it will pull up all the samples that corresponds to that particular barcode. As I mentioned, as label printing, I can define what fields I would like printed on the labels. And these are very special labels because they can survive up to absolute zero, absolute temperatures in the freezer. So this is a special printer, a special label. And any of these fields that you see here, we can print out to the label printer. There's also an extensive reporting field. I showed an example here that is just showing the depth of how customizable reports can be. So if you'd like to find everyone with a tumor after the 11th of June 2022, who is above age 35, who is female, you can do that. And you can have any combinations of advanced searches and filtering in STAMS, which is quite useful, especially if you have really, really large number of samples and many, many freezers. There's also other reports that are available. So you have a sample audit trail, everything is tracked. Patient reports, you have samples reports, you can get summaries, and then you can also get information on external distribution. So if you were to ship samples out or receive samples, you can track those in the system also. This was a new feature that we added because we realized that tracking fluids and tissues were not enough, but there was a significant demand for a brain repository and how to manage brains. So we developed this capability here in collaboration with the DoD and also NIMH. This is the first iteration. So we have the case intake. We have a set of different tabs. In intake, we have the protocol number, patient name, basic information about the brain when it has arrived. And for case history, we have more in-depth information about the person who has donated the brain. We also have the ability to help the pathologists when they're doing their grossing and staining. So in real time, they can go in and check what kind of stains, what kind of grossing, what kind of processes are they using for the brain that has been acquired. And they can actually print a report for this. And there's also the histology information for the brain. So how many slices, what kind of stains, how was it fixed? We also are able to track because brain donations, a lot of them happen by shipment. So we can track when a shipment has arrived and if any items are being sent out. And this is the next of kin interview. We have built this in. So a lot of times when we get a brain donation, the brain repository individuals will call up the next of kin to get information on the donor. So we can get medical history, any psychiatric history, family history, those kinds of information. And we have it here. And again, this data is searchable. So with a large enough repository, you can find all the brains, all the patients with specific criteria, such as traumatic brain injury or depression, and then look at the corresponding brain slices. In our analysis tab, what the pathologist had asked for was the ability to view the different brain images, and then what they wanted was to be able to make notes. So when a brain comes in, it gets sectioned and sliced, and then it goes underneath the microscope for very high resolution images. So these images are about a couple of gigabytes each. and they're very high resolution. So the pathologist will view these and then create their pathology notes for each item. What we're trying to develop next is the ability to annotate and also mark up the images. And that's a next step in this brain analysis tool. We also have the ability to generate reports. So this is an example of a neuropathology report. So the system will pull all of the information you see here, clinical summary, grossing description, all of these from fields within the system. And then it creates this report that the head of the pathology department can then sign and then keep it on file or send it to whoever needs that. Very important because someone has donated their brain. We usually send a letter to the family to say, hey, this is, what we have found. So we have a template here, and then within the system, again, we pull like the patient's name, we pull any of the pathology notes, findings, and then the system actually generates a pretty nice templated report, and we print this out and then we mail it to the next of kin or the family. With that, I would like to thank the team. They've done a phenomenal job. I also want to thank all our users and contributors and all the working groups, because a lot of these features we took months to develop based on working groups that had met repeatedly and given us information requirements and also refinements over time. Are there any questions for the STAMS module?
I think, Dominic, there was one question. I think you addressed it, but you can take a look and see if you can just, or can you see the questions, Dominic?
Yes, I'm seeing a parent-child relationship management.
Yeah, it looks like Andrew felt that you did answer it, but
Yes, so we do have that tracking, and then for each child specimen, there is the parent. There's a label that says parent, and then also there's a label. So let me go back to the presentation. I'll pull it. So right here, so you have sample parent history and then remaining samples. And if you click on it, it would give you that information that you can track. So if you pull the audit trail, it will show you which one is the parent, which one is the child, and then what's like the remaining pieces. Any other questions for STAMS?
Yeah, so hi, this is Stefano speaking. Do you know whether this module, I'm sorry I missed the beginning of this talk, but do you know if this module has been used to track IPS lines? Because there are many parent-child samples, and then quality control has to be done at each step. So it's a little bit more intricate.
That's a good question, Dr. Maranko. Not yet, but we are, again, trying to gather more requirements, and we can definitely build that feature in.
Okay, thanks.
yeah, the one nice thing about the BRICS application and also the applications that Dominic is working on, that we have all the source code, so we can update and modify where we need to. So lots of flexibility there.
Okay, thanks.
So I think we're ready for phase 2, Dominic.
All right. So the next module I'd like to talk about is the clinical trials management system. Again, I have no conflicts of interest. All the views here are my views. Don't blame the government for anything. It's all me. I'm the bad one. So BRICS consists of a set of different modules, as I mentioned before, and today I'm going to talk about the clinical trials management system. So what is a clinical trials management system? Why do we need one? Well, doing a clinical trial, there are a lot of different moving pieces. So the first piece is study management. We have to look at accruals, how are the subjects doing, study timelines, are milestones being met. If it is a new drug, new device, what is the status? We have to manage, if it's a drug or device, manage the pharmacy, giving it out, administering it, and then also track how much of these were like given out. What is the monitoring status of the clinical trial? Has any adverse events been found? The data safety monitoring group meetings, how were the outcomes? And then also personnel management as people roll on, roll off the study, what are their roles? And then are there any publications or analysis and findings? In terms of study documents, all of us are well aware doing research that research generates a lot of documents. There are regulatory documents, IRB reviews, approvals. If it is a long enough study, you might have different versions of consent forms. And then we have to do all those trainings and certificates and make sure that those are up to date. Lastly, we have to generate reports for the sponsors, for reviews, for monitoring. There are a lot of different reasons we have to generate reports. Currently, these can be done, but a lot of these things tend to live either on individual computers, SharePoint drives, or there's a need for multiple systems and then having to sit down and reconcile information across multiple systems. So what we're trying to do really is, again, have an integrated solution so that everything lives in one place. If personnel roll on or roll off, it doesn't affect access to information, and then there's better continuity for the study and for the PI. So at the NIH, there are two really big systems. We hear about this a lot. So the IRB system is called Protect. Protect actually was implemented on January the 17th, 2023 to replace IRIS. So this is the platform for submission of new studies and ongoing actions for IRB review. The other system is ProTrack. So ProTrack focuses on clinical center reporting and activities. What ProTrack does is keep track of information. So ProTrack is not new. It used to be known as PQS, but then it got renamed to ProTrack. So it keeps all the funding information, data information, recruitment status, accrual, any clinical trial specific information. That's what ProTrack keeps. So what we have done is integrate the CTMS in BRICS with the ProTrack system so that we can pull all of this information into CSTAR or BRICS essentially without having to retype and re-enter all that. So how does that look like? On the main page, we have protocol information. So things such as, the IRB protocol number, study title, research type, FDA regulations, all of this information we can pull from ProTrack right into the CTMS. Study personnel, who are all these individuals, we can also pull this information from ProTrack. So we have their roles, how many hours are they working on a specific study, and then what is their affiliation. Scientific review information, we can gather this, and we're in the process of trying to connect to Protect to pull this from the IRB. But I was actually informed that ProTrack actually has this information also. So we're working on trying to pull this automatically. But we can keep track of all of the reviews, when was the meeting, and then what is the outcomes? Was it approved, not approved, pending review? We can keep that here. And if there are any documents, we can also add that to this scientific review page. Same thing with IRB review. Again, we can keep all of our documents here. So it's in a central location and it's accessible and it's also tracked. Any FDA information, if there's sponsor information, IP information, if there's FDA correspondence, a lot of times for clinical trials towards the middle or the end, there will be FDA correspondence, so we can track that information here. And then any sponsor documentation, if it's internal sponsors, external sponsors, any collaborators, we can also keep copies of those documents and then track the status. We also, key important thing is having a data management plan. We try to encourage all of the investigators to have a data management plan and a statistical analysis plan, because it really helps with the study. And if things are not, if the study goals or objectives are not being met, they can go back and always revise these. But having a written and accessible data management and statistical management plan helps, especially if it's a multi-year study. And then monitoring. This is where we would have, depending on the frequency of monitoring, if it is quarterly, biannually, any of the monitoring results and findings will be here. And usually the first monitoring visit starts with a site initiation visit. That's kind of like the kickoff to the start of the clinical trial. And the outcome for that, usually we try to, again, have one central place where we can record all of these monitoring outcomes. If it is an investigational new drug, as I mentioned before, we want to track pharmacy information. We want to have the pharmacists be able to go in and see what they are giving out if they're unblinded. If they are blinded, then we are able to configure the platform so they can only see the minimal information, but still be able to keep track of inventory and also who has been administered the specific drug. In terms of drug accountability, we want to be able to track the manufacturer information, the lot number, and then any like drug information. And depending on the clinical trial and the drug, some of these tend to move pretty quickly. Like, for example, the manufacturer might say, hey, we've come up with this new finding. We would have to change the dosing or change the drug. So we want to be able to keep track of that, especially as the drug is being developed. Data safety and monitoring board. This is really important, especially if there is an adverse event or a non-compliance event. If there's an adverse event or serious adverse event, the data safety and monitoring board would have to be convened right away, and they would have to review what has happened and determine if the trial can continue or not. So the outcome of the data safety and monitoring board has to be documented in writing and has to be kept for FDA review when the trial is being submitted or when it has concluded. Typically, we would have a list of data safety monitoring board members, their roles, the contact information, and then there's also charter information as to what exactly is the role of the data safety monitoring board, what will each members do, what are they allowed, and their roles and responsibilities. Every meeting information has to be documented and also recorded. And then we also have this central location for statistical analysis and manuscripts. And again, it's just a convenient way of tracking what progress or what outcomes or what findings have come out from this specific study. We can also generate other reports like adverse events. Our adverse event generator now is integrated with Medra and also CTCAE for grades. We also have non-compliance reports. And a big thing here is we have training records, licenses, annual review. As my colleague Priti had mentioned earlier in the presentation, in the account module, rather than having a lot of these trainings and documents outside, we can actually track the trainings, records, and licenses within an individual account. And as these expire, the system can actually send an email to remind the users like, hey, your training's expiring in 2 weeks. You have to update these. So again, we have integrated this into BRICS. I'd like to thank the team. Again, it's a huge lift and everyone has done a tremendous job supporting us. And again, I'm grateful to our users. Our CTMS user group met for about 1.5 years, and they gathered information from across the NIH, across all users. And we also had a DoD CTMS user group that met for 2 years. And I really applaud the collaboration between the two groups in coming up with the requirements and then refining them and then helping us develop the CTMS module. Are there any questions?
So that was really a lot of great information, Dominic. This is functionality that can be deployed with any instance of BRICS. That's correct, right?
Yes, sir. Yes, sir. That's correct.
So, and the neat thing, and for some people who here might be a little bit new to the BRICS system, it's not one system. It can be a system that is set up to support multiple users or projects, or it can be set up for individual projects, so that if it was deployed, say, to support, here an example is for traumatic brain injury, FITBER, which is designed there's specific modules and functionality that's to support traumatic brain injury, or there's another instance that's to support Parkinson's disease biomarker program. And one of the neat things is that we've also built this into kind of a mesh that we can connect these systems together when appropriately, and to be able to share information and be able to search and find cohorts across different instances. So that actually also, provides some additional kind of functionality to know that some of your data, if there's comorbidities, that you could then associate that, again, where appropriate and where appropriate credentials and authorizations have been provided, that you could share and collect data. So we have a question here. I don't know if you can see your Dominic can see your chat. We have a question from Andrew. Are you still here, Dominic?
Yes. Sorry. So that's a really good question, and the data itself. So let me take one step back. So BRICS has the ability to collect data in CDISC format and in formats that the FDA wants. The important caveat is we have to set up the data elements prior to start of the study. If a study has already started and they are migrating into BRICS, then we have to do a mapping, and it depends on how those original data elements were set up, because if you don't have all the fields, then it's difficult to go back and recollect. So yes, in short answer, BRICS does have the capability to send data to the FDA in the format that they want over.
And if you want to come off and join the conversation, Andrew, just feel free to unmute yourself.
Yeah, hi, Matthew. I guess I don't have any further questions.
Yeah, I think.
Yeah, I mean, it was
okay.
candid. I thought the answer was yes. I mean, that's why you're doing it. But just to kind of understand the nuance there. And Dominique, you're right. Of course, you got to have it right going in at the beginning.
Yeah, and just to reinforce that, so if prospectively you're setting up a study, you set up the data elements with the common data elements within the data dictionary, and so you're right, you're then collecting that data consistent with CDISC, and then it becomes very easy to export that data in an appropriate format for ingestion to the FDA. So if there are any other questions, this will be the last presentation for the summer, and we'll start back up in September. But always feel free to reach out to one of us, email one of us, and we'll be happy to touch base with you over the summer. There's lots of really interesting work that we're performing at the time. We're adding some AI functionality. We've done a lot of prep work for that in setting up the data so that the data is more AI ready. And again, that's in our data dictionary. We're adding UMLS coding. We built a tool to help us support the UMLS coding for data elements. Another feature, and Preeti may have touched on this, is our data dictionary can now output the data elements in NLM endorsement format, so that when you export the data elements and you want to get endorsement on those data elements through NLM and the CD governance process, they'll be very much closer to the state to get validated through that process. So again, we're trying to work closely with different groups here at NIH to make the whole process much more seamless, and it'll be easy to exchange either data or information about that data more effectively across the different stakeholders. So with that, I'm gonna Stefano,
Yeah,
you have a question?
so you mentioned data preserved in UMLS format or something like that. What does that mean?
It's a unified markup, unified, I forget what the actual acronym is, but it's a semantics that allow you to add concepts to data elements. So if there are multiple data elements that could happen in a data dictionary because of lots of projects, that you can then semantically assign them the same UMLS code. And great, Allison added the link there. And so it also allows it to make the data more AI ready. So if you go and extract the data, so we can programmatically query into the system, get access to the data. Again, you have to have proper credentials. And we also provide the data elements for all the data that was collected in that particular query. You can then, instead of trying to figure out what those data elements are, you can semantically look at the UMLS code for that data element and say, oh, that relates to ethnicity. And then you can automatically process that data. We're also adding, good, Robin added the acronym there. Unified Medical Language System is what UMLS stands for. But so what we're trying to do is provide the ability to programmatically process that the data more effectively, and also cross-map the data. So, you know, all the common data elements or data elements within a particular system like FitBIR, we try to reuse data elements in other systems like PDBB, but sometimes they use them and use data elements in a slightly different way. But semantically linking that through the unified UMLS coding helps, again, aggregate that data and then also possibly automatically process that data.
So now we create the clinical summaries that are kind of that are written in plain language, so to speak. Would it be possible with this UMLS system to read all the symptoms, for example, that are listed in these narratives and database them automatically?
UMLS would not necessarily help with that. So there's some large language models or AI techniques that would help to parse that data and then to build data elements that might support that. You know, and I say that with caveats. And, you know, there's been some work in doing that, and it does, you know, it's going to get it right, and I'm not going to give it a percentage, but more often than not, but it's not going to be a perfect solution. We are working on some AI and large language model techniques or tools that will support finding data within our system, both at the common data element level, the metadata level, and then eventually to the data level.
Is this seminar, do we have access to the recording of this seminar?
Yes, so if you go to the BRICS website.
Okay, great. Thank you.
Yep. Any last question? We've got time for one more question before summer break here, if anyone has any last question. And I don't see anyone's hand raised or anything in the chat. Again, we want to thank everybody. Have a great summer, and we'll hopefully meet up again in September with some nice new functionality. Okay, have a great summer, and talk to you soon. Bye-bye.
Thank you, everyone.